In the introduction to the activity, the authors want to make sure that we have a common vocabulary that they want you to focus on. And one of the ideas is that we can take groups of animals and break them up into taxa. And taxa are just literally groups that have similar characteristics. And there are things like, as we've discussed already, kingdom, order, class. But there's also just the idea of just grouping them based on phylogenetic groups, or groups that have evolutionary relationships, or groups that have inherited characteristics from ancestors. And so when we're talking about phylogenetics, we're talking about looking at not necessarily just common characteristics, but looking at things based on the most recent common ancestor. I think you all understand this, but I just want to make sure that we make that point clear, because it's not just as simple as just grouping them into similar groups. It's similar groups, and then we consider, are these groups related somehow through some kind of genetic inheritance? And that's where we get to similarity that we call synapomorphy. Synapomorphy is important because synapomorphy looks at this idea of similarity that is based on shared derived characteristics. Now, again, derived means that it's from a common ancestor, but that common ancestor is recent. So it's recent ancestry. So we look for the most recent common ancestor, and most recent common ancestor, MRCA. And we try to compare it to the outgroup, which is going to be a group of organisms that are somehow related, since we start off with the assumption that all living things on the planet are related in some way genetically. And so based on that assumption, there's a group that doesn't share a specific characteristic that is a distant or ancient, has ancient characteristics that are shared with a much older common ancestor. And in the example they give you here, they talk about fish having jaws, and that lampreys don't have jaws, and maybe not having jaws is an ancestral trait, where all the group of fish that have jaws have a more recent common ancestor. So lampreys are then therefore our outgroup. And again, that outgroup is our comparison group, or our control group, that we use to say these are the ancient traits, and these are the traits that this taxa, right? This taxa shares with one another. So these traits are recently derived. They have a more recent common ancestor, and they're due to more recent evolutionary steps or evolutionary events. And then at the bottom we talk about, as we all know, the cladogram, and how we can use a matrix of characters, right, to create a cladogram. That drawing, that graphic organizer that allows us to look at how things are related evolutionarily. What are the ancestors and how are the organisms within a taxa, within a group, within a clade, how are they related to one another? So here we have this, the character matrix you're supposed to do research on and fill in, and this is very difficult for a lot of people. There was good reason for it, because some of the things are questionable, like molting. What do we call molting? Remember, we said that for molting we're going to talk about arthropoda, right? It was a yes. And we also said that mistakenly on this chart it should have been also nematodes, right? Nematoda should have also been a yes on that. And you have the corrected version the physically correct, the corrected version that's has been handed to you, but this electronic version was not corrected. Also, radial cleavage. We know that yes. Let's see, where is it? Or rather, radial symmetry exists for Cinderia, but remember, Cinderia has bilateral symmetry when they're young, but they end up having radial symmetry when they're mature. So that pretty much fills in, this chart here fills in all your missing steps here, so identifying the characters that you had to fill in. Here you see the chart of the organisms that we had discussed earlier. Here's lamprey without a jaw. Notice it has vertebra. The vertebra is an ancient trait that defines the group. But the group that we're talking about is a group with jaws, and the shark has jaws, but and fins, where the pantheroid gland only belongs to this clade here. And at each intersection, each common ancestor group, we can define a clade or taxa, right? That we can say, okay, these guys share something. They share keratinous scales, and where these groups, these three here, share amniotic egg. And this is very powerful because it helps us to organize groups as we need to look at where these traits fall in and how closely are these organisms related to one another. So we can take a look at this, and we can see that from the previous chart that how many different states you can have for any particular character. Most of these characters only have 2 states, a yes or no, but some might have 3, and some might have the option of not applying. So you have to be careful with traits, and as you saw from previous examples, often they'll use pluses for yes and negatives for no. Also, they'll use B for bilateral and R for radial, so they'll use letters or variables to represent character states as well. So you see in this simple example that you can, and they talk about it here in the paragraph just above it, that you can identify quickly that only 2 of all these organisms have these scales. Okay. So obviously these 2 share a trait that the others don't. Of course, the question is, is this a convergence event? In other words, are they really related, or are these 2, do these 2 have these scales because this one, the crow, evolved it separately from the garter snake? It's possible under the same conditions, right, that 2 species can derive the trait. The derivation of a trait can happen twice, so we can have this convergence evolution occurring. Or is it that they're actually related? And we've done the kind of math that's required to kind of look at the similarities. We've added up the derived traits, and we looked at what's probably the best cladogram for a particular matrix, right? So let's take a look at what they came up with. So here in this character matrix, as you can see, or that character matrix, as you can see, was put into this cladogram to explain the similarity between crow and garter snake. They put it in their own clade, saying that a common ancestor shared these types of scales with them, rather than. And remember, we picked the most parsimonious, or the most the simplest of answers. So if these were separated, if this one were here, for instance, and the other garter snake here and crow here, then because we think garter snakes are closer to frogs than they are to crows, it would require this evolutionary event to happen twice. And we say, one of the things we try to remember is the simplest answer is probably the best answer. So the most parsimonious or simplest answer is that these 2 are related to one another, and that these scales evolved once, not twice. So here, after looking at it yet one more formation, one more possible tree, right, phylogenetic tree, they're asking you to take that character map and the character states from your chart and place them in a cladogram as best you can. Okay, so, all right, so to place these characters where they fit best in this hypothesis, and remember, a cladogram is a hypothesis, and all depends on the evidence that we have. We're detectives, and we're placing the evidence where they needs to go, and we're making our decisions logically based on the facts that we have to make that decision by. And these are your facts, the data you collected. So the Hox gene, there's only 3 organisms on this cladogram that have it, and that's Annelida, right, has it. And so that means that the Hox gene had to have developed here, and the other 2 are mollusca and platyhelminthes. And mollusca is here, right? And platyhelminthes is here. So there's one possibility, there's 2 possibilities, right, when you come to a question like this. One, that the Hox gene evolved here and here. So that's 2 evolutionary events of the same, so a convergence event. Or it happened here, and all 3 would have had it, except the Nematoda lost it. So, which one occurred? We can't tell. But both are equally right because both take 2 evolutionary events to occur. So it really wouldn't matter which one you chose. So we can say Hox happened here, and it happened here as well. So maybe Hox is just one of those evolutionary events that's really favored to occur under certain conditions, and it just happens. Or it could, again, the other option would have been that it happened here, the evolutionary event happened here as well. That would have been two evolutionary events that led to the development of Hox, and then one that meant that Nematoda lost it. That would have been 3 total. These are 3 evolutionary events as well, so they're equally they're equally likely. All right, so the next is the type of skeleton, chordata, annelida, and arthropoda. And if you look at the type of these 3, each one has their own type of skeleton. So you have one is an endoskeleton, one's an ectoskeleton, right? And one is a so you have a hydroskeleton, ectoskeleton, endoskeleton. So each one of these had to develop its own type of skeleton in this hypothesis. So this would be endo, and this is ecto or exo, right? Oh, there it is. They're already placed. And so let me get rid of those. The endo and exoskeleton is already placed here. Here's exo, and here's an ecto or exo, and here's endo. But annelida would have had to have perhaps the H, maybe, since a lot of these, let's look at Cnidaria and Synapomorphy, and let's look at, if we look at Cnidaria, Synapomorphy, right, as an endoskeleton, they put it on already. So the hydrostatic skeleton is all the way down here, and so everyone else where these events didn't occur have a hydrostatic skeleton, and that's what you see here. This group developed an endoskeleton, and the periphery, where are they? Oh, wrong one. Sorry. These don't have an endoskeleton. It's a mistake on the chart. But in any case, anything that doesn't have the changes has a hydrostatic skeleton. Okay, so moving on to segmentation. you see that there's the first three, these three all have the same, all have segmentation. These two don't, right? These two don't. So this one doesn't, and this one doesn't. And in fact, everyone else doesn't except for these three. So really, that's just one event, and that one event was segmentation, and that happened here. And that's easily done. So we've done the Hox gene, the type of skeleton, segmentation, and we've identified a possible error here, right? Now notochord. Notochord is Annelida, is no, right? So this is no notochord. No for arthropod, but chordata, yes. In fact, no one has notochord here. And this is a mistake for sure, since, well, no, that's true. All right, so notochord, no one has it except for chordata. So there again, notochord would be placed here. And that checks off notochord. This type of larvae is only found in two, right? No one else has it. That's Annelida and Mollusca. And really, the only way that's going to happen is if we put TL here and TL here. It's two events. Otherwise, it'd be just all kinds of reversals going on. So that takes care of the larvae. Elongate cylindrical body. Everybody has it except two. That's the chordata and the periphery. So that leads loss of, right? And there's a loss of okay, so that takes care of that. Molting happens only in arthropoda corner. This is also a mistake. Molting also happens, I didn't fix it. Molting also happens in nematoda. So arthropoda nematoda. So molting, again, must have developed twice throughout the evolutionary history. And radial cleavage, only two of them have it. It's chordata and echinodermata, or I can't pronounce this word. Echinodermata and chordata are the only two that have it. So again, this here we have the development of radial cleavage, and here you have the radial cleavage. So this has us really working on a lot of different evolutionary events to make this hypothesis work. Multicellular, everybody's multicellular, so symmetry, only one has radial symmetry, right? And here we have it. And only one has two tissue layers, and there it is there. So when we count the number of different evolutionary events that would have to happen to make this hypothesis work, right? So this is key. When we look and count how many different evolutionary events had to occur, we count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 events, 19 evolutionary events for this hypothesis to work. Is this the most parsimonious? Is this the simplest? is this the simplest explanation? Would this give us our, our most parsimonious cladogram? And that answer is yet to be determined because we have to look at the other hypotheses. Now, this hypothesis, as you can see, it's different. The hydrostatic skeleton is still there. The endoskeleton happened here. Already I can see that these two events only happened once. Endoskeleton here and exoskeleton here. Everybody else has hydrostatic skeleton. Already this is more parsimonious, but we'll have to see if it overall is most parsimonious. Let's look at the Hox gene again. Only Annelida has the Hox gene, right? And Mollusca and Platyhelminthes. So Platyhelminthes, Annelida, and Mollusca all have the Hox gene, and look what you can do here. Now remember, the Hox gene was all over the place in the other hypothesis, where in this hypothesis I only have to have it evolve once. The type of skeleton here, as you see, it's already placed. And again, I think, this is a mistake, should have been an H. So here there's a type of skeleton, and you can see that, again, it's already placed, and you only have 3 events occurring. Segmentation, there's only the first 3 have segments, so so again, we're talking about Annelida, Arthropoda, and Chordata having it. So Annelida, Chordata, and Arthropoda having segmentation. Now here, unfortunately, we have to have segmentation happen 3 times in order for it to work. Now, notochord only happens once, and that's with Chordata. This type of larvae, once, twice. So we're talking about Annelida and Mollusca. Annelida and Mollusca to get So we're looking at here is where we would develop this type of larvae. Done. Elongated cylindrical body. Everybody except Chordata and Periphery. Everybody except Chordata and Periphery. So Porifera, Proifera, oh my God, Porifera and Chordata, those 2 are the only ones that don't have elongated bodies. Okay, so that takes care of that. And sorry about that. My phone is just going crazy today. All right, so molting only happens once. It actually should happen twice. Again, I didn't fix it. Molting happens in nematoda as well. So it happens twice in arthropods and in nematoda. So molting happens here. Oh, look at that. It happens here, and so if you put molting here, only evolved once. And radial cleavage only happens in Chordata and in ectodermia. So radial cleavage only happens once here. Excellent. Multicellular, everybody has it. Symmetry, only have radial symmetry in one, which is here. And tissue layers, only one has 2 tissue layers. So how many of these do we count here? We count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So 15 versus 19, this is obviously more parsimonious. So right now, with the evidence that we have, and maybe some of this evidence is wrong, or maybe we collect more evidence, but whatever. Right now, based on this evidence, we only have 15 here and 19 in the previous hypothesis, so this is the hypothesis we're leaning towards. Now go ahead and do this hypothesis. Analyze it, place the characters from our matrix the way that, place the characters in our matrix as best you can, and see how many changes, how many evolutionary changes would actually occur if you use this particular hypothesis instead of the previous one. And so if you use this hypothesis, place these character traits, remembering that there was 2 mistakes that we identified, and that's that molting happens in nematoda for sure, and this is a hydrostatic skeleton on endoskeleton. So those 2 are mistakes that are obvious, and then there might be some other mistakes, but in any case, you have to keep using the same matrix and the same to compare all 3 hypotheses, otherwise it won't work. So whatever changes you make, you always keep making this, you make sure that all 3 of the matrices are the same when you're comparing them to your hypotheses. So go ahead and place these characters the way they are for Hox gene, just like we did in the previous 2 examples. Place them where they go here, and then count how many total. Make sure you're placing them based on where they fit, okay? All right, good luck. We'll go over the other parts of this in another video.